Hello everyone. Uh, Today we will have session number 31 on paper 2 biochemistry, which is part of the second semester. So in this session we will be discussing about the general introduction of the enzymes. So this is what we have discussed already in the introduction paper. Yeah. So what are enzymes? So enzymes are biocatalyst that increases the reaction rate without being used up. Without being used up. Without being used up. Most enzymes are globular proteins. However, some enzymes are RNA molecules. Uh, these RNA molecules behave like catalytic tools, catalytic RNA, or sometimes even we call them as ribozymes. The study of enzymatic process is one of the oldest field of biochemistry, dating back to the year 1700. The study of enzymes has dominated biochemistry in the past and continues to do so. So now let us look at the history of enzymes. The term enzyme was coined by uh, Kuhne in the year 1878. So the name actually enzyme comes from these two words en and zyme. En means within and east. Literally, it is something within the east. So enzymes were first identified in the east. Uh, because of the most recognizable reactions, popularly known as alcohol fermentation by an enzyme called as zymase in yeast. So hence we uh, get the name as enzymes. So even till today it is in use. So actually it means En means within, zyme means yeast. So within the yeast. So this is how the term enzyme came into picture. So these are some of the major contributors in the field of enzymology. Uh, so, uh, so we have Dubrufont, uh, who prepared malt extraction. Payen prepared an enzyme, diastase. So we also have the contribution of Schwann. We also have the contribution of Jones and Jacob, who recognized with remarkable foresight that the catalytic nature of the enzymes. Pasteur demonstrated alcoholic fermentation. Pasteur demonstrated alcoholic fermentation, and James Sumner at Cornell University isolated and purified the enzyme urease from jack beans. Now let us look into the difference, how an enzyme is differing from the catalyst, how the enzymes are going to differ from the catalyst. Even we have lot of catalyst in chemistry. So like we use nickel, we use some other chemical compounds that enhance the rate of a chemical reaction. Then now the question is how these enzymes are going to differ from the chemical catalyst. The enzyme do not alter the chemical equilibrium point of a reversible reaction, but only uh, they enhance the speed of the reaction. So this is what the enzyme does is. And one most important thing is the enzymes, all enzymes are protein, and they are being colloidal in nature, and they are confined only to the living world. So they are present in plants, they are present in animals, they are present in microbes, right? uh, and catalyst, they can enhance the rate of many reactions, but most enzymes are very specific in their mode of action. They either act on a single or at a particular group of compounds. So they are very, very specific in their action. Enzymes are highly specific in their action. So this part we will see in properties of enzyme. So where we can, yeah, this is the another example where I can tell you about how this biocatalyst have a upper hand when compared to the inorganic catalyst. So when we look at the type of reaction being catalyzed by the enzymes, most of the enzymes, they have greater reaction specificity. That's what I was telling. And they avoid side products. They avoid side products. And most of the enzymatic reactions, they occur at pH 7 and 37 degrees centigrade, which is very common, which is very normal temperature, or which is at very milder conditions. They don't require a higher temperature, or they don't require some special kind of conditions. Higher reaction rates in a biologically useful time frame, capacity in a biologically useful time frame, capacity for regulation, they control a number of biological pathways. Yeah, now let me come to an enzyme. An enzyme is composed of only proteins, is known as a simple enzyme, and some enzymes are complex enzymes which are composed of protein and relatively small organic molecules, is known as a complex enzyme. The protein part of the enzyme is called as the apoenzyme or a apoprotein. So this is an apoenzyme or an apoprotein which will be inactive, and the other non-protein part is also called as a cofactor. So this is a cofactor. So this together, apoenzyme or apoprotein and cofactor, together we call it as an holoenzyme. The cofactor, so this particular guy can be an organic molecule, or it can be an metal ion. It can be organic molecule, or it can be an metal ion. So this is very important. Let me explain it again. The structure of the enzyme. The enzymes can be either, so we all know that enzymes are basically, they are biocatalyst. They are proteins in nature. Right, and they are highly specific in their reaction. So when we look at the structure of the enzyme, so this is the most important core part of an enzyme, called as apoprotein or apoenzyme. And this protein part will be inactive, and when it is going to associate with the other component, that we call it as an non-protein part, that is a cofactor, then the enzyme becomes a holoenzyme or a complete enzyme. So this cofactor can be either, a metal ion, or it can be an organic molecule. In many cases, most of the organic molecules, like vitamins, they act as coenzymes. So this particular thing is called as a complete enzyme, and this is the pocket where the substrate is coming and binding. I hope it's clear. Yeah. So this is the catalytic cycle of an enzyme. The enzyme is available with an empty active site. So this is your enzyme, and the substrate, say for example sucrose, comes and binds here. The substrate enters the active site, which enfolds the substrate with and induce fit, and then what we can see is the substrate is being broken down, and it is being converted into the products. The products of sucrose are fructose and glucose. They are being broken down. A disaccharide is being broken down. So the reaction catalyzed here is the breaking of the glycosidic linkage, or a glycosidic bond is being broken. So this is how the cycle of events that are going to occur in an enzymatic reaction. So first we have an enzyme, then substrate comes and binds. See, usually the enzyme is a large molecule. When compared to the substrate. The enzyme is quite large, the substrate is quite small. The substrate comes and binds to a particular area. So this particular area, or this pocket, we call it as an active site of an enzyme. Active site of an enzyme where the substrate binds either by induced fit or by any other mechanism. It is going to bind here, and then the substrate is being catalyzed, or the bonds are being broken, and it is being converted into the product. So at the end of an enzyme substrate reaction, what we get is we get enzyme as such, and we get enzyme product. We get enzyme and product. So the idea here is, so at the end of the reaction, the enzyme is not going to change. It is going to remain as such. Okay, I hope it's clear. Yeah, now let me come to the another important aspect that is called as nomenclature, or naming of the enzymes. So naming of the enzymes are generally named according to the reaction they are going to catalyze, say by fixing a suffix. Say for example, we have a class of enzymes that are going to act on lipids. Automatically here the enzyme will be lipase, LIPAC. So here we are going to add ASC, which is a suffix, and it is going to get converted into the product, and we get the enzyme at the last. Later, this particular system was being modified with the International Union of Biochemistry and Molecular Biology. They developed a new kind of nomenclature for enzymes. So each enzyme is described by a sequence of 4 numbers preceded by EC. So every enzyme has EC. So this EC stands for enzyme commission, and this will be followed by some number like this. Say for example like this. When classified, each enzyme is assigned the EC number, the form of digits separate by periods. The first number categorizes the enzymes based on its reaction. Yeah, this is an example of how what I was telling. Yeah, so this is an example for an enzyme. Say this is let me take, this is EC 1.2.3.4. So here EC is common for every enzyme. EC stands for enzyme commission, and the first number 1, it is for the class. So we have 6 classes of enzymes. So you say 1, 2, 3, 4, 5, 6. So this we are going to discuss in the next lecture, and say lecture number 32 will be discussing classification of enzymes. The first class of enzymes are oxidoreductases. So 1 stands for oxidoreductases. 2 stands for the subclass, acts on the aldehyde or other group of OX group of the donors. 3 is the subclass, oxygen is an acceptor, and 4 is the serial number 4 is the 4th enzyme classified in this particular subclass. So this is how enzymes are being named. Yeah, I hope you have got the point, and these are the references I have used for preparing my slides. So in today's class we have discussed about, let us have a quick recap. We have defined what are enzymes, and so enzymes are basically biocatalyst. They enhance the rate of the reaction by decreasing the energy of activation. So enzymes are basically proteins. There are some exceptions where some RNA, they have catalytic activities. We call them as ribozymes, and the term enzyme was coined in 1878 by Frederick Kuhne, and we have also seen how a biocatalyst is having an upper hand or a greater advantage when compared to the inorganic catalyst. We have also looked into the structure of the enzyme. So enzyme has a apoprotein. Enzyme has a protein part and a non-protein part. The protein part is called as apoenzyme, and the protein part is called as a coenzyme or cofactor. So if it is a organic molecule, we call it as a coenzyme. If it is a inorganic molecule, we call it as a metal ion. So the enzyme will be usually inactive. When it binds with the cofactor, it becomes active, and then it can catalyze the reaction. And we have also seen the cycle of enzymes. So in an enzyme substrate catalyzed reaction, the enzyme binds with the substrate to form to form an enzyme substrate complex, and this enzyme substrate complex gets converted into enzyme and product. So in this reaction, at the end of this particular reaction, the substrate is being broken down into product. Rather, the enzyme is going to remain unchanged so that it can bind with other substrate and can continue the cycle. We have also looked into the nomenclature. The nomenclature is being framed by the International Union for Biochemistry and Molecular Biology. So according to this, there are 6 classes. So this is how the nomenclature is being done, and these are my references. Thank you very much. If there are any doubts or questions, please uh, put in the comment section. Please like my videos, subscribe my videos. These PPTs and videos will be followed by study material, and other literary materials will be shared to the students. A effort has been made to reach out the students during this pandemic. So I thank you one and all for being part of this.